Hello and welcome you again to my webinar, All You Need for Immuno-Oncology B-Cell Research or Solutions for Immuno-Oncology and more. Let me first give you a quick overview of the agenda, what I'm going to talk about today. First, I will give you introduction to the company. Then I will talk about B-cells and antibodies in tumor immunology in general. Afterwards, I will talk about the products and the solution Miltenyi Biotec offers for B-cell research, for the cell separation, cell analysis and cultivation. Last but not least, I will talk about the applications and the examples for immuno-oncology B-cell research. So here I will give you the example of therapeutic antibody generation. So let's start with the facts about Miltenyi Biotec. The company was founded in 1989 in Germany. We have 14,000 products available worldwide. And right now we have more than 55,000 cell therapies that have been produced with Miltenyi Biotec products. We have more than 2,000 employees, whereas our staff is consisting of 44 different nationalities. Yearly 2,500 patients are treated with cell products using Miltenyi Biotec technologies. And in 73 countries, our products are sold via distributors and in 28 countries via direct sales. We are a very strongly R&D driven company, so we have 475 employees working in engineering, clinical research and R&D. So let's start with our whips of the day, the B cells. So B cells play physiologically an important role in the human immune system. So of course they play a role in antibody production and memory function. They also play a role in cytokine production, wound healing, antigen presentation, the balance of Th1, Th2 cells, dendritic cell regulation, tumor immunity and co-stimulation. So here you see a tumor cell with the tumor antigens on the surface. When a therapeutic antibody binds the antigen, it can induce programmed cell death, or a therapeutic antibody can bind to soluble factors, which is a process that is called neutralization. And by the binding to the soluble factors, this functionality of the soluble factors is blocked. Another thing that can happen is that therapeutic antibodies bind to specific receptors so that they block their functionality. For instance, EGF receptors can be blocked, so growth factors cannot bind anymore. So another mechanism of action is the activation of the complement, which is called complement-dependent cytotoxicity. This leads ultimately to the formation of the membrane attack complex and the lysis of the cells. And one very important mechanism is the effector cell recruitment. So the effector cells bind via the Fc receptor to the Fc part of the antibody. This bridges together the target and the effector cell, which results in killing of the target cell. So NK cells can kill tumor cells via a mechanism that's called antibody-dependent cellular cytotoxicity. Macrophages can do antibody-dependent cellular phagocytosis, and T cells can do T-cell dependent cellular cytotoxicity. So always research aims to have antibodies of maximal therapeutic efficiency, and therefore antibodies can be engineered. Such antibodies can be checkpoint inhibitors, bispecific antibodies that have two different specificity, bridging together effector or target cell, blocking antibodies that block inhibitory receptors on NK cells, and also antibody conjugates. These are actually antibodies that are coupled to biologically active cytotoxic agents or drugs. So now, after we know how and why therapeutic antibodies can be used in cancer therapies, let's talk about the research products Miltenyi Biotec offers to help you to do such research. To simplify our constantly growing product repertoire, we compiled our products in groups. For instance, we start with the sample preparation, which is the first step of the so-called workflow. We have here kits, consumables, and reagents for the sample preparation. The next step is the cell separation. There we also offer instruments and consumables, kits, and much more to separate your cells. After you separated your cells, cell culture is following. So we have different media, cytokines, and many stimulation agents for that. After we have our cells, we also want to analyze them. So for that, we also have flow cytometers, a cell sorter, software, antibodies, kits, and other reagents. The newest addition to our world is the imaging and microscopy. So here we also offer consumables and instruments. But actually, why should you choose Miltenyi Biotec products? That's very simple. Just select the best. Here I have some pictures of cells that were isolated with MUX microbead technology, and here of a competitor product. And you directly see that there are many beads in the microscopic picture visible. And also here in the electron microscopic picture you see the big beads on the surface of the cell. But when you look onto the picture of the MUX microbead technology, you don't see the beads. And this is due to our column technology, because the MUX column amplifies the magnetic field by 10,000 times. This means we just need minimal labeling of nano-sized beads for cell separation. The beads in the column matrix have a very hydrophilic coating, and they have a large space between the steel spheres. This allows a free flow-through of the cells through the column and allows a very gentle enrichment of the cells without any unspecific activation of the separated cells. Also, this allows that we have a lot of free epitopes on the surface of the separated cells, and this allows the full downstream compatibility, such as flow cytometry. Our beads are used in clinical research for the manufacturing of cell therapies for more than 55,000 patients. That means our beads are also FDA approved. But now let's come to the B-cell products. So you can isolate human B cells from many different starting materials. You can isolate them from whole blood, from other blood products just as Buffycoat, LRC, and Leukopak. You can isolate them from PBMCs or from bone marrow. So here's an overview of the B cell subsets you can isolate using our kits from PBMCs. So you can start by broadly analyzing and separating your pan B cells, or you can directly go into specific subsets of B cells and isolate naive B cells, plasmablasts, plasma cells, memory B cells, and abnormal B cells. On the right-hand side, you see that we have for each subset and need a different solution. But no worries, you don't have to remember all these different products. You can always check on our homepage about the products and also for the other starting materials. So one of our newer technologies is the MUX Express technology. In comparison to other technologies, it is much faster. So you can isolate your B cells within 20 minutes from whole blood. Other technologies need much longer. Only the centrifugation step of the density gradient centrifugation needs 35 minutes. And when you then use a competitor technology to separate your cells, it needs another 40 minutes. That means you can save more than 55 minutes in each separation step. And for this technology, we have available a pan B-cell isolation kit, a B-cell isolation kit, a BCLL cell isolation kit, and anti-ROR1 microbeads. Another technology that saves you a lot of time for isolating your B cells is the straight from technology. Here you can isolate your B cells, CD19 and CD138 B cells from many different blood products, such as whole blood, Buffy-coat, leukopak, LRC, but also from bone marrow, without the need of any density gradient centrifugation, what saves you a lot of time. Here I have some exemplary data for you for the straight from whole blood CD138 microbeads in comparison to a competitor product. Here you see the starting material that was gated on CD45 viable cells. They were stained with CD19 and CD138. In the upper panel you see the result of the straight from whole blood CD138 microbeads that were subsequently stained for CD19 and CD138. And you can nicely see in the dot plot your positive isolated plasma cells. In the lower panel, you see the cells that were separated using a column freeze system. The massive labeling of the cells leads to epitope saturation, which prevents an efficient staining of the cells and leads to a very poor result in flow cytometry. So you don't really know after separation if you really separated your plasma cells. So far we only talked about human B cells, but of course we also have products for mouse B cells. So we have products for the isolation of B cells from spleen and lymphoid tissue, from body cavities, and also from bone marrow. So here we also have an overview of B cells you can isolate from spleen, but most of the products can also be used for lymphoid tissues. So here you can analyze subsets like marginal zone and follicular B cells, regulatory B cells, here in detail B10 B cells, also germinal center B cells, plasma cells, memory B cells, and you can broadly separate also your pan B cells. Here on the right hand side you see again all the different products, and again you don't have to remember them, or you can always check our homepage, and there we also have the products for the other starting materials. So as I told you before, Miltenyi Biotec is a complete workflow provider. So after you separated your cells, you of course want to analyze them to see what you got. So here we offer the flow cytometers, antibodies, kits and reagents. And this is very important because B cells have so many different subsets, and so it's important to really have good tools for cell analysis. And therefore our reaffinity antibodies have the best features. That are recombinantly generated antibodies that provide superior lot-to-lot consistency and purity. They all have the same IgG1 subtype. That means you just need one isotype control. To eliminate the background signal during your analysis, the Fc region was mutated that abolishes any binding to antibodies from the antibodies to the Fc receptors, of course. So we have many different antibodies available where you can analyze your B cell subsets with. But I just put together a nice panel for you, how you can analyze some of the subsets with just 6 different recombinant antibodies and 1 viability dye. So with that, you can analyze your B cells. So first, the cell debris, doublets, and dead cells were gated out. And then you can analyze plasma cells, memory B cells, and transitional B cells. When you then have a closer look on the plasma cell gate, you can analyze immature plasma blasts and mature plasma cells with the markers CD20 and CD45. When you then check the memory B cell gate, you can further analyze them for switched memory B cells, non-switched memory B cells, and in the very same gate you can also analyze your naive B cells with the markers IgD and CD27. So here I have some dot plots for you. That is exactly the strategy I told you before. The B cells were purified using straight from Leukopak CD19 microbead kit. And you see here the plasma cell gate, the transitional B cell gate, and the memory B cell gate. Here is the further analysis of the plasma cells. So you see the mature plasma cells and the immature plasmablasts. And by further analyzing the memory B cell gate, you see here the switched memory B cells, also the non-switched memory B cells, and the naive B cells. So after we separated and analyzed our cells, of course, we want to culture them. So also we have a lot of products for B cell cultivation available. We all know that for most of the B cell studies, B cells need to be expanded and activated. For that, we even have a kit available that have all the stimuli you need to get activated and expanded B cells. The kit is consisting of a CD40 ligand multimer, a cross-linking antibody, IL-4, and a xeno-free media. The cross-linking of the CD40 on the surface of the B cell leads to cell activation and proliferation. And when IL-4 is binding to the IL-4 receptor on the surface of the B cell, it also leads to effective activation and proliferation, as IL-4 is a known growth factor for activated B cells. But we additionally have a lot of other cytokines. We don't just have IL-4, we have IL-21, TNF-alpha, and many other cytokines for human and mouse tissue culture. We also have stimuli like TLR ligands or the FLT3 ligand, and some of the products are also available in GMP grade. If you want more information about our tissue culture products, please check out our homepage. We're going to talk B cell activation. You actually need 3 signals. So first signal is the antigen binding to the B cell receptor complex, seen here. The next is the co-stimulation that's happening from activated T cells, in detail T helper cells, that are getting activated by binding with their T cell receptor complex to the MHC2 complex of the B cell, which is presenting a peptide of the antigen. And here the stimulation is happening when CD40 ligand of the T cell is binding to CD40, and a certain signal that is happening is the cytokines that were produced by the T cell, the activated T helper cell, and those bind to the cytokine receptors on the surface of the B cell, what leads to full activation, clonal expansion, and differentiation of the B cells. And the differentiation actually depends on the cytokines that were produced by the T cells to the B cells. Okay. So now you know that the co-stimulation of the T cells is a very important mechanism for B cell activation, proliferation, and differentiation. So now you see why we built this B cell expansion kit with the CD40 ligand multimer, the cross-linking antibody, and the IL-4. And here you see the expansion rates of the B cell expansion kit. When you use the B cell expansion kit, you can get up to 10-fold expansion of the B cells in vitro within 14 days. IL-21 has also been reported to be an effective stimuli for the B cell activation and proliferation, and therefore we tested to replace actually the IL-4 by IL-21 in the culture. And we observed that the B cells have an earlier onset of expansion, and this is probably just due to the IL-21, what's really nice. So we could prove by that the B cell expansion kit is a standardized and reproducible system that also can be used for clinical processes requiring GMP standards. So now let's come to the actual applications and examples. So to the development of therapeutic antibodies. So for the development process of therapeutic antibodies against cancer, human and mouse donors are needed. So this could be patients suffering from cancer or mouse tumor models. When you work with solid tumors, first the tissue needs to be preparated. So for that you can use the GentleMax dissociator and the tumor dissociation kits. After you have your single cell suspension of the tumor, your B cells can be separated using B cell separation products. For this purpose, I would highly recommend CD19 microbeads because the positive selection offers the best features for such applications. In the next step, you of course want to analyze your separated B cells. And this I showed you already in the cell analysis section. So here you use reaffinity antibodies, via dyes, and the MAX-Quant analyzer. So after you analyzed your cells, you of course want to culture them, expand and activate them for downstream purposes. For that you can use the B-cell expansion kit and or other cytokines and additives. So downstream applications could be screening for specificity and functionality of the antibodies or also sequencing. Because when you get your genetic information, you can recombinantly express your monoclonal antibodies to do further testings on them. But also you can do expression profiles, you can analyze the subsets, you can also discover new targets for CARs or new checkpoints. You can find dominant clones in the tumor, or you can check a complete B-cell repertoire of a patient. Of course, such processes can also be performed on blood products as a starting material, and that's especially happening in humans. This has been done, for instance, a lot in breast cancer studies, where the complete B-cell repertoire of breast cancer patients was compared to healthy individuals. So now let's discuss some pain points that can occur in the cell separation step of this process. When I'm recommending positive B-cell separation to our customers, one pain point I always hear is the unspecific activation of cells where the people are scared of. In this scheme here, you see actually how a positive separation procedure is done. You label your B cells with the magnetic beads, and then you transfer your cell suspension in the column. The cells that are not labeled run through the magnetic field, and the cells that are labeled are getting held back. You can elute the magnetically labeled cells by taking the column out of the magnet, and you get your highly pure and happy B cells in that step. And we did a lot of experiments to check if the cells are getting unspecifically activated by the separation process. And we found out no matter what you do, positive or negative selections, the cells are not getting activated. And this is an experiment we did for that. So we analyzed B cells after the separation and after the cultivation for 7 days in the presence and also in the absence of activators. On the left-hand side, you see the results for the pan B-cell isolation kit, the negative isolation kit. And on the right-hand side, you see the release CD19 microbead kit, the positive selection. And you see here are the cells directly after separation for the negative isolation kit. Most of the cells are double negative, and that looks just the same for the positive isolation. Also, after cultivation for 7 days without any stimulation, the cells are still double negative, not activated. Just when you add your stimuli, your additives, the upregulation of the activation markers is happening. again for both of the kids. We also did some comparisons to alternative column-free cell separation products. The cells were also analyzed by flow cytometry after the cultivation, after the separation, and also again in the presence and in the absence of activators in the B cell culture. And you can see that the B cells purified with our column-based technology showed no activation in the culture when there are no stimuli in the culture. But when you separate your cells with a column-free technology, you can really see the upregulation of the activation markers, even though there is not any stimuli in the tissue culture. Another pain point in this step can be the B cell integrity. For sequencing, of course, the RNA quality is very important, especially when you want to have a complete patient repertoire that should be screened. And for that, we showed that the purification process does not affect the cellular or the RNA integrity of the cells. So this provides the best conditions for sequencing, also for single cell sequencing. Here the B cells were purified using straight from leukopak CD19 microbeads. The total RNA was extracted from the purified B cells, and the RNA was analyzed with a microchip-based electrophoretic run, and it showed a very high integrity and exact best quality of the RNA with RIN values of 9.9 to 10. So let's come to the next step, the cell analysis. So what are pain points that you could face here? Pain points might be the low reproducibility and inconsistent results, or high background and unspecific binding. To get such nice plots as we got here, reaffinity antibodies, biodyes, together with the max quant analyzer are the best solution. The recombinant expressed antibody provides the consistency and very easy handling. You just need one universal isotype control. Also, they prevent unspecific staining and high background thickness. And what is really nice, no FCR block is required. Together with the max quant analyzer express modes, the analysis is almost done on its own. So now let's talk about the pain points for the B cell cultivation. This is a point where a lot of challenges can occur. Of course, when you have your precious cells and you culture them, you of course don't want to lose some of them. You want to have the same subset distribution so that your sample is really representative. So also you don't want to have unreliable B cell activation or an inefficient expansion. And all this is a huge challenge. So when you use the B cell expansion kit, your B cell have a similar distribution of naive B cells, non-switched B cells, switched B cells, and plasma cells compared to the original fraction when you culture them. Here you see the examples after 14 days of culture, and you see a reduction of the naive subsets for the unstimulated cell and also for the stimulated cells, and the same distribution of memory cells as the original fraction. And this is due to the fact that just naive B cells don't have such a long survival in vitro as memory B cells. And that's just normal. And very interesting is that when you add IL-21 to the culture instead of IL-4, the whole repertoire is shifted to a more maturated switch status. So you see a higher number of switched memory B cells and also a higher number of plasma cells compared to the result for IL-4. And this is just due to that IL-21 is considered to be a plasma cell differentiation driver, while IL-4 is described to be important in B cell survival. The B-cell expansion kit is a standardized and reproducible system, so it can be used in future for clinical processes, also requiring the GMP standards. And combined with the max premium grade cytokines, you can optimally expand and activate your B cells, because we have an exact dosage with the units per milligram, which allows a very high reproducibility. So you don't need to worry about inconsistencies anymore. So for more information, you can always check the Miltenyi Biotec homepage. We always provide new materials to always keep you posted. If you want to have more information about the B-cell separation and analysis, you can write in the chat that you want to have the PDF of this poster, where it shows you all the separation of different B-cell subsets from many different starting materials, so the stuff I couldn't show you now. And that's it so far from my side. I thank you very much for your attention,